Welcome, everyone. This presentation, Preparing Samples for Microscopy, has been broken up into 5 parts to make it easy for users to watch. We encourage you to watch all 5 parts in the order that we have them so that you can get the full benefit of the presentation. And while this is primarily aimed at users or potential users of the imaging course optical at the University of Arizona, the principles are widely applicable. Before we begin, I want to bring your attention to the quote here at the bottom of the screen. It says 90% of good imaging happens at the bench, not on the microscope. And this is from a colleague, Dustin Higashi, who was here at the University of Arizona. He's now at Oregon Health Sciences, last known. And Dustin was and is an outstanding microscopist, and some of his images have been involved with on journal covers, so he knows what he's talking about. So let's get started. One of the things that we want you to do is to plan ahead. We see too many people that come to us with preconceived ideas and they haven't thought ahead. If you consult with your core facility before you start, you'll avoid mistakes of spending money on things that you didn't need. The unfortunate truth is that your faculty lead or your postdoc or your lab manager may not actually have the expertise you need for certain types of experiments. So talk to us. We'd be happy to work with you. And the truth is, even experts routinely consult with one another. We talk to other experts when it's outside of our field of experience. It's also important to know what instrumentation you have access to and what it can and can't do. There is a joke in the microscopy world that the answer to 90% of questions is, it depends. And the University of Arizona Microscopy Alliance has a comprehensive listing of the microscopy core resources on campus. It's a great place to start, but it's also a place to meet the people who can help you decide whether that instrument's going to serve your needs or not. And you can use the contact form on the Alliance or reach out to specific core staff for more help. One of the issues is to make sure that you have a good protocol. Unfortunately, the truth is that some published studies and protocols are sloppy science, and they will be a waste of time and resources. So just because it's in a journal doesn't mean it's any good, unfortunately. Many well-loved and handed down lab protocols have errors. And you really need to have a proven, reliable protocol for sample prep. And how can anyone do reliable science without one? One of Stephen Covey's 7 habits is begin with the end in mind. So know what you want to do with the samples up front and plan accordingly, or look backwards in a sense and try and figure out what it would take to get there. And unfortunately, with some of these things that you find out there in the scientific literature, it's appropriate to trust but verify. It's important to gather your reagents. Have everything that you need. Make sure that they are fresh. They are similar lot numbers. One of the issues that has been shown is that cell culture media often vary a great deal between lots. And if you're working in a lab and someone else made the aliquots, were they someone that was fairly reliable in doing the dilutions? And how many times have the aliquots been in and out of the freezer? That unfortunately can really beat up fluorescent labels or antibodies. Some potential hazards you want to be careful of, just physical hazards. Make sure you have the safety equipment that you need. Is there a first aid kit around? Do you know where it is? I've seen a lot of first aid kits where the Band-Aids are at least a decade old, and even the little paper around them doesn't stick anymore. Learn about the risks of the chemicals that you're working with and have the appropriate chemical safety materials that you need. Know how to get rid of your waste as well safely. Be aware of the fact that there are some biohazards involved with certain types of experiments. And if you are bringing BSL-2 samples, certainly to our core or really any core, you should coordinate with the core facility in advance. And if you're working with laser-based systems, there's often training involved that's required to comply with federal oversight and things like that. It's true, it does matter to plan ahead, and experience has shown that people that really pay attention to these picky details of sample prep get better image and more reliable image data. If you've ever seen any of the microscopy contests like the Nikon Small World, those pictures are amazing, but they're using the same microscopes in core facilities that you are. But the difference is the people that end up taking those high quality pictures, in addition to having interesting samples, also have an understanding of how to get the most out of their sample using proper sample prep. And just as a following or an ending note, the NIH and NSF have a big concern about rigor and reproducibility, and that's an issue that we want to be careful of in the world of microscopy as well. Just a quick acknowledgement of where these images came from.